Good day, everyone, and thank you for joining this Visionix webinar. We are pleased to have with us Professor Pablo Dighiero, MD, PhD, a renowned expert in ocular surface pathology, corneal disorders, and dry eye disease. Professor Dighiero serves as president of the Association of Dry Eye Centers and the World Interventional and Dry Eye Society, and currently practices at the Barbizon Ophthalmology Center in France. With a distinguished academic and clinical career, Professor Dighiero is internationally recognized for his contributions to the understanding and management of ocular surface diseases, particularly through the integration of advanced diagnostic technologies and individualized patient care. Today's topic, 2025 Innovations in Solix OCT for the cornea, ocular surface, and dry eye. This presentation will explore the latest updates and clinical applications of the Solix OCT platform in the evaluation of the cornea and ocular surface, particularly in the context of dry eye disease. So with the evolving capabilities of multimodal imaging, including epithelial mapping and corneal topography, we now have powerful tools to better assess and monitor corneal changes, leading to more precise diagnosis and personalized treatment strategies. Professor Dighiero will guide us through these innovations, supported by clinical case examples. So if you have any questions during our webinar, please feel free to email us at contact@visionix.com, and our team will get back to you as soon as possible. Professor Dighiero, thank you for being with us today. We now hand over to you.
Thank you very much for this invitation to this webinar, in which I will talk to you about the applications of the OCT Solix for exploring the cornea, the ocular surface, and dry eye. This webinar will take place in 6 parts. In the first, we will discuss the importance of dry eye in ophthalmological diagnosis, how to conduct a quality dry eye examination, and why the Solix is useful in these cases, how the Solix allows for evaluating the effectiveness of modern dry eye treatments, a new application of the Solix, which is topography, is included in the exploration of the cornea, and we will see the ease of use. This will lead us to the use of the Solix for preoperative and postoperative follow-up of patients operated on in refractive surgery. And finally, I will show you some images of the evaluation with the Solix of corneal pathology. For 5 years, we have created 2 scientific societies aimed at improving the management of dry eye. LACOS, the Academy of Cornea and Ocular Surface, aims to establish the best diagnostic methods and protocols to achieve optimized care. The WAITS, which is the World Intervention and Dry Eye Society, aims to evaluate the best current treatments, how to combine them with each other for optimal management of dry eye. So the 3 essential missions of the WAITS are to have an accurate and reproducible diagnosis of dry eye. And for that, you will see that we will rely on a score called ITAO. Establish a rational and thoughtful therapy for the treatment of dry eye, and finally exchange and incorporate the best international experts into these objectives. Why dry eye? Currently, at least 10% of the world's population is affected by this condition. That still represents nearly 1 billion individuals worldwide who suffer from dry eye, and for the ophthalmologist, almost 1/3 of their consultations, aside from glasses and refraction, are involved in dry eye. That is to say, it is the primary reason for consultation for a frontline ophthalmologist. So how can the Solix help me make this diagnosis of dry eye and evaluate it? The score we created, called ITAO, to properly assess dry eye, focuses on 4 essential points. the state of the cornea during the lack of lubrication in 4 stages. The T for telangiectasias and ocular inflammation, which allows them to be rated similarly in 4 stages. Meibomian atrophy in 4 stages as well, and meibomian obstruction in 4 stages. What we have learned in recent years is that meibography has become an absolutely essential tool for managing the diagnosis of dry eye. Over the past 10 years, I have had the opportunity to test all the meibographers available on the French market, and I was pleasantly surprised to find that the Solix was not only a good meibographer, but it also provided me with plenty of information on the condition of the cornea, thanks to the epithelial pachymetric maps. So let's discuss the topic of meibography with the Solix. As you can see with one of my assistants, the infrared measurement is very, very simple and quick, right? You see that in less than 5 seconds, we have the infrared image of the meibomian glands. I always pair it with the color evaluation of the conjunctival and bulbar surface. You can see it here. So similarly, in 5 seconds, we have these 2 pieces of information. We always do the 4 eyelid, so here we will image the upper eyelid. With a bit of practice, blipping the eyelid is very simple. And you see once again that we can have a well-defined and good quality meibography. So all this to say that in less than 5 minutes, you have a complete meibography with a color evaluation of the ocular surface. Here are the results we obtained for this patient. You can see on the right the meibography images in both infrared and color. On the left, you have the topographic maps, the epithelial pachymetric maps, and by simply pressing the small CT or topo button, you can immediately switch from topography to, on the video, we see the image of the epithelial pachymetric maps, and by pressing the small button instantly, you have the corresponding topography for this patient. So all of this is remarkably simple and quick, which is extremely practical for daily examinations. The quality of this meibographer allows us to have the classification into 4 classic stages for meibomian atrophies. So stage 1 that we can see in green with the green background where there is no atrophy, stage 2 where the atrophy is between 25 and 50%, stage 3 with atrophy between 50 and 75%, and stage 4 atrophy greater than 75%. And you can see that the Solix provides you with all this information very precisely. The evaluation of telangiectasias also allows them to be classified into 4 stages: no telangiectasia, superficial telangiectasia of the free edges, superficial conjunctival telangiectasia, or deep stage 4. The corneal epithelium, which is the third aspect for evaluating dry eye, you can see it here in 4 stages. The first stage is a normal epithelium. The second stage is an epithelium with a false hyperplasia, mostly due to tearing. The third step or the third stage is superior atrophy due to rubbing, and the most advanced stage is diffuse atrophy of the corneal epithelium. So here are several stages that you can see represented at the same time, and all this is a mechanism caused by the rubbing of the upper eyelid on the top of the cornea, which gradually atrophies it from the top towards the center in the most advanced cases. So be careful with false hyperplasias. As I mentioned, most of the time, as we can see on these slides, it's simply an excess of water, or sometimes patients who apply eye drops that remain on the edge of on the surface of the cornea. An important element to accurately diagnose during the surgical dry eye assessment is what we call lead wiper epitheliopathy, which is due, as you can see on this slide, with the fluorescein that stains the free edge of the eyelid, and this thickened free edge will rub against the upper cornea and create this epithelial atrophy, which is so important to diagnose during management. As with mammographies, in recent years, I have been interested in all devices capable of measuring the corneal epithelium, and I have found that the Solix is extremely reproducible and reliable, and probably one of the best, if not the best, on the market for evaluating epithelial pachymetric maps. So the goal was to analyze the 4 essential points of dry eye. You see that with just the X, the E, the T, and the A are perfectly evaluated, and the O is thus evaluated with a clamp. Meibomian, no device in the world currently does it automatically. And so whatever the device, the Solix allows you to diagnose 3 out of 4 items. So the Solix, we use it for diagnosis and obviously also to assess the effectiveness of treatments. You can see here a patient with stage 4 meibomian atrophy, the most severe that we started to by pulse and UMS. You see that in July 2021, we had quite a diffuse epithelial atrophy. After a few months, this atrophy began to be resorbed, and thus on the differential maps you can see on the right, we clearly see the favorable evolution of the corneal epithelium thanks to the various treatments. The same goes for tearing. You can go from stages of false epithelial hyperplasia to normal epithelium after treatment. This very interesting case of a patient we saw for the first time in June 2022 with, as you can see, the epithelial pachymetry maps extremely disturbed on the right by significant tearing, and on the left, huge epithelial atrophy. And thanks to the various treatments in August 2022, we already had a good improvement, and see that by March 2023, we had managed to restore a good quality epithelium. For the case of lead wipers, as I told you, they are the cause of the upper epithelial atrophy. You can see here on the slide from June 2024 with a significant lead wiper, and after treatment in July 2024, the improvement on the lead wiper eyelid, which gradually led to an improvement of the upper corneal epithelium. Topography with the Solix, that's one of the major advancements of recent months or the past year. It's extremely simple. Once again, you have the epithelial pachymetric maps. You just need to press the little topography button, and there you can have all the topographic maps you want, anterior, posterior, and you even have indications on corneal labirometry. So all that in 1 second. So this is really extremely useful in daily practice. Why is it useful? Because, for example, you sometimes have patients you want to quickly screen for keratoconus. And you see here in the images, corneal epithelial with the classic donut, that in 1 second, by pressing the topography button, you have the topography maps that perfectly confirm the diagnosis. This makes screening, diagnosing, and monitoring keratoconus extremely simple. Here is another image of a more advanced keratoconus, where it was important to also perform meibography with the Solix, since we can see atrophy of the meibomian glands, causing dryness that led the patient to have an irresistible urge to rub their eyes, which is what we know to be a formal contraindication in keratoconus, as rubbing can promote the progression of the disease. We compared the topographies of the Solix with that of the Pentacam HR, and we have perfectly superimposable results, as you can see on this slide. And so now we need to get used to using these progression pachymetric maps, like for glaucoma or the retina, which we call trend analysis, and we can do it with the corneal epithelium, with topography, as well as with the total corneal pachymetry. In refractive surgery, what is the purpose of the Solix? I use it systematically. Initially, it was for the corneal epithelium. Then, when we started doing mammographies, it provided us with very useful information, because in a case like this, for example, a 25-year-old patient who wanted to undergo LASIK for myopia due to the significant atrophy of the meibomian glands, we preferred to recommend PRK, which went very well, and the patient is doing fine. We also use the Solix and topographic maps for patient follow-up. Here you see a patient before and after surgery, and you can clearly see the stromal pachymetric maps and epithelial pachymetric maps, as well as the corresponding topo. So very easy and quick to do for the follow-up of operated patients. For patients suffering from dry eye after LASIK, which is currently the primary complication of LASIK, you see that we can look at the depth of the LASIK flap. This was a patient operated on in the 2000s with microkeratomes, with blades, where the flap depth was often a bit greater than what we currently have with the end of the second. And so we were able to monitor the patient both in pachymetric maps and in topography, and the mammography revealed that the patient was suffering from dry eye due to meibomian gland dysfunction, as you can see on the right slide, where there is significant gland atrophy. Similarly, these very interesting patients coming for post-LASIK dry eye consultation, you can see on the mammography a stage 4 atrophy, so a very, very significant stage. And on the epithelial pachymetric maps, we had something very interesting. Obviously, the central part corresponded to the Alzheimer treatment of the cornea, but also on the periphery, these epithelial atrophies, which indicated the problem of dry eye due to friction. Similarly, for LASIK complications, we can see here, for example, a case of epithelial ingrowth, where with LASIK we can perfectly measure the depth of the deposit for long-term monitoring. Corneal pathology is thus consolidated into two main categories: complications related to dry eye in order of frequency, as you can see here, from rare nular filtered keratitis to catarrhal infiltrates to Salzmann's nodules to Kogan's dystrophy and to superficial punctate keratitis. Here is an example of the information provided by the Solix to evaluate the nummular infiltrative keratitis. You see the deep subepithelial deposit that disrupts the architecture of the anterior cornea. The catarrhal infiltrates that we can perfectly diagnose here, and which also allow for follow-up after treatment. The Salzmann nodules, the information provided by the Solix is important because it allows us to see the cleavage plane in which the nodule is located in relation to the epithelium and Bowman, which helps determine the type of treatment that will be proposed. A more advanced case of Salzmann with two nodules, and you see on the right image that with the OCT in front, we can assess the depth and spread of the nodules. The most common complication among the corneal conditions is Kogan's dystrophy, and here you have some examples, and I now use a lot of end-face OCT images to assess the extent of Kogan's lesions and thus see which ones can benefit from PTK treatment. Another case, you see, it's extremely simple to do, very practical and very useful for illustrating the problem and educating the patient on the possible treatments we will propose. This case is very interesting because you see on the image of the right eye a cogane that has never been treated with these central epithelial disturbances that will alter visual acuity. And in the image on the left, you see that the center of the cornea has been cleared because this patient was able to benefit from a PTK. The corneal pathology that is not related to dry eye, I have given you some examples, like the lattice dystrophies that we can see here. On radial images, we can have the exact depth of the deposits. And on the frontal images, you can once again see the spread of the lesions to best propose a treatment, which can be either PTK or lamellar grafts for this type of patients when visual acuity is extremely altered. Our common case is Tagerson's keratitis, which generally causes quite significant epithelial atrophies. And see here between the before and after, how the epithelium improves after treatment. In this case, it was cortisone. And this Tagerson keratitis can also, as seen in the image below, be evaluated in EnFace OCT to count and monitor the lamellar deposits of this keratitis. Something extremely important that I would like to convey as a message is that patients treated for glaucoma with eye drop treatments often experience significant toxicity from the eye drops, causing them ocular surface inflammations. And see here this patient with severe glaucoma treated with a triple therapy, who presents major epithelial atrophies due to the toxicity of the eye drops. And so, as I was saying now, the glaucoma monitoring charts must include epithelial pachymetric maps because the appearance of epithelial atrophies should lead you to modify your diagnosis, and most of the time, the indication for an SLT laser will arise. Similarly, for this patient who is being monitored for intravitreal injections, anti-VEGF, you see that after a few injections, the epithelium starts to suffer greatly, probably due to the betadine used for disinfection, causing these quite troublesome epithelial atrophies, which will cause significant discomfort for the patient. So just like for glaucoma, now the follow-ups for retinal pathologies must include epithelial topographic maps to assess the toxicity of treatments. We are reaching the end of this webinar, and I would like to convey 5 important messages to you. The first is that you now have, with the Solix, a comprehensive tool for evaluating dry eye, and you therefore do not need, for example, to invest in a mammographer, since you already have everything on the device. The Solix is essential for a good preoperative refractive surgery assessment, and especially to detect the 2 major contraindications of LASIK, which are dry eye and keratoconus. You have seen that in less than 1 minute, we are able to screen for these 2 conditions and thus disqualify a patient for LASIK if there are abnormalities of dryness or keratoconus. The pachymetric maps are really excellent, and I use them every day because of their reproducibility, which helps me evaluate a lot of corneal pathologies. As I just told you, glaucoma and AMD during their follow-up must necessarily include epithelial pachymetric maps to properly assess the toxicity of treatments on the ocular surface, particularly the corneal epithelium. And we had the pleasant surprise of seeing that the topographic maps from the Solix were comparable to those of the best topographers on the market. For those interested in all these pathologies, we organize a conference in Paris every year. This year it will be on December 12th and 13th for continue discussing all these topics. Thank you for your attention.
Thanks a lot, Professor, for this insightful presentation. I just have a few questions for you before to finish. The first one, should the Solix be used as a screening tool or reserved for specific complex cases?
So as you could see now, due to the image acquisition speed in Solix, we can simultaneously evaluate the cornea, stromal, and epithelial pachymetric maps. And with the tools in front, you can do everything. So I would say that the Solix can range from ultra-fast screening to exploring the most complex pathology. And that's what is extremely practical in the daily evaluation of these issues.
Okay, great. My second question, is corneal end-face OCT analysis expected to become a routine examination?
Yes, for me, it already is. As you have seen in several clinical case examples, we use it daily because of the quality of the images, which complement other images, whether they are inline or tachymetric maps. And so, yes, for me, it's already something essential in routine.
Okay. Now, is performing an additional scan such as epithelial OCT on the Solix compatible with daily clinical practice when monitoring patients with glaucoma or AMD?
Yes, more than important, it's essential. Now, I think that serious monitoring of a patient treated for glaucoma or treated for AMD without a corneal topography map or an epithelial pachymetric map would be a mistake, because you have seen all the information it provides you. And so for me, it is almost as important, I might be exaggerating, but almost as important as the follow-up maps for glaucoma and the retina.
Okay. And can the Solix detect early epithelial toxicities related to glaucoma treatment before clinical signs appear?
And yes, that's the whole point. That is to say, the only X will start showing you epithelial atrophies even before the patient eventually starts complaining, which allows you to adjust your treatment. And as I was saying, in the case of glaucoma, for example, to start considering SLT laser to replace the drops and their toxicity. And for the intravitreal injections, for example, to have less aggressive disinfection protocols when the corneal epithelium starts to suffer.
My last question, could the Solix become a gold standard for monitoring epithelial damage induced by anti-glaucoma and intravitreal treatments?
Yes, because after testing almost all the devices that exist, we can consider that the Solix is an extremely efficient device for these epithelial pachymetric maps, not only from one patient to another, of course, not only reproducible within a few minutes apart, but perfectly superior to many systems of tachymetric maps, for example, which are only on 6 millimeters. We will not see all the problems, for example, that appear between 6 and 9, which are all the problems of superior epithelial atrophy. So indeed, I could no longer do without the Solix to get all the information provided by the tachymetric maps.
Okay, thank you. So on behalf of Visionix, we would like to sincerely thank Professor Dighiero for his insightful presentation. Today's session has showcased the versatility of the OCT Solix as a truly multimodal platform. We highlighted the value of the epithelial mapping feature in managing dry eye, glaucoma, and AMD, particularly in understanding the impact of treatments on the ocular surface. We explored the integration of the new corneal topography module, as well as the comprehensive suite of tools for the detection and follow-up of cornea diseases. And finally, we discussed the ETAO score and how Solix enhances diagnostic precision and clinical decision-making, ultimately supporting better patient outcomes. So please don't hesitate to email us at contact@visionix.com for any questions, and our team will get back to you as soon as possible. Thank you so much for being with us today. It's been a real pleasure to share this session with you, and we truly appreciate your participation. We look forward to connecting with you again soon. Until then, take care and have a wonderful day. Thank you.